lesser GIT irritation. So let us move on. But now it's not very loud. Already done that. So, next, the previous one. So the Z isomer is active, lacks a CNS side effect, introduction of a floral group. It's active. I don't know whether this is. The analgesic is overshadowed by concern that we already mentioned, and we already said is active. It's a prodrug, it's active form. Is a pseudodite which has a long active. The filler is out of play. That one we had already done.
But check your email, please.
Then we can note the isosteric replacement of the Indo ring with the Indian ring system resulted in a derivative with therapeutically useful anti-inflammatory activity and fewer CNS and but with other undesirable effects that we've already said, poor water solubility and resultant to crystalluria. The replacement of the N-parachlorobenzoyl substituent with a benzylidine function resulted in better molecules, better derivatives. However, when the 5-methoxy group of the Indian isoster was replaced, the fluorine atom enhanced analgesic effects were also found. So there was need to replace the methoxy group. The methoxy group with the fluoro group, it resulted in better, it improved on the activity of the Sulidac compared to the Indian indomethacin derivative. The decreased water solubility of the Indian isoster was alleviated by replacing the chlorine atom of the phenyl substituent with the sulfonyl group. We've already mentioned that. So we can't be able to explain why there was a change from the indomethacin Indian isoster to Sulidac to have the fluoro group to have the chloro group replaced by a fluoro group, a methyl sulfonyl group, because that was necessary for optimal activity and also necessary for minimal toxicity. Clinically, it has only about have the potency of indomethacin in treating inflammation and reducing fever, but it's equipotent in analgesic effect. It's absorbed as the inactive sulfoxide. It causes the fewer GI disturbances, no prostaglandin biosynthesis inhibition. This is what I was asking you. It's absorbed as the inactive sulfoxide. Remember, it is changing the body to the active sulfide. Therefore, no prostaglandin biosynthesis inhibition in the stomach. Sulidac is a prodrug and is converted to a metabolite that appears to inhibit the cyclooxygenase system approximately 8-fold as effectively as aspirin. So it's more active than aspirin. The thioether metabolite is longer lived than the parent. The thioether is the SCH3, which is the one which is more active than the sulfoxide. Both the indomethacin and Sulidac assume similar conformations that we have already said. They must be non-coplanar. The two rings must be non-coplanar, and the Z or cis isomer is the isomer that will be responsible for activity. So there is a lot of similarity there. We are only saying there is a change of the substituent. Number 5, change of the substituent paraposition of the previously indomethacin. There is also change of this nitrogen replaced by an Indo ring, replaced by an Indian ring with good activity, but a slight reduction in anti-inflammatory activity. Pharmacokinetics. That is for indomethacin. Absorption occurs rapidly on oral administration. Being an acidic substance, bound. And of course, all drugs that are protein bound will also be responsible for a lot of drug interactions. It is converted to inactive metabolites. When you look at its structure, you see it can undergo 5-O demethylation at position 5. It has a methoxy group, so it can undergo 5-O oxidative demethylation to give a hydroxyl group at that position, and that, of course, resultant metabolite is inactive. It can undergo conjugation with glucuronic acid. Remember, we did mention that all these drugs have a carboxylic group and therefore the carboxylic group is a site for conjugation. Then non-hepatic enzyme system, hydrolyzed indomethacin to end deacylated metabolites. We are going to look at that. So it can also undergo it can also undergo hydrolysis of the amide, whereby the indole ring and the other benzoyl moiety will also be free of each other. That's this what we're talking about. Yes, with indomethacin. So here is our structure of indomethacin. So the first here you have a whole OH here, meaning this is oxidative O dealkylation. Oxidative O dealkylation, where you have removed the methyl group there, you have a hydroxyl group there, everything else I believe is the same. Then this also can undergo glucuronic conjugation. You have a carboxylic group there, glucuronic conjugation. This can undergo amide hydrolysis, this here, amide hydrolysis, and that will be our product. This compound here also, even after oxidative and demethylation, it can also undergo hydrolysis to give us that, and also that compound can also undergo glucuronic conjugation. So the main methods of metabolism is glucuronic conjugation, amide hydrolysis, and oxidative O dealkylation for that drug. And we can be able to get to derive all those routes of metabolism after we know the structure of the drug and we know the various routes of metabolism. So that is what we are looking at. Then we have again our Sulidac here. I didn't bother drawing its structure. Sulidac. And the first Sulidac is something. Which one have we started with? That's a fluoro group. Sulidac. Oh, I think this one, I think we have just shown. This one is glucuronic conjugation there. Which metabolism have we done here? I think this is oxidation. This is an oxidation. This is oxidation. So that we get, I think this is CH3. This structure has a slight mistake. It's O. See, must be something a bit fishy there. Because there was Sulidac. So here we have this. Well, that's what the other one, which is what is happening. This group here, see, it becomes CH3S double bond O. So it is the sulfone. So I think there was something. Let's go back. So it will be CH3SOO. I think that's correct. You have to check and see the correct structure. It is CH3SOO. So these two O's are actually on the sulfur, double bond O and a double bond O. So that is the sulfone. So that's an oxidation, and that compound is inactive. That's the sulfone. And then also the sulfone can undergo glucuronic conjugation, still inactive. Remember, the oxidation, the oxidized metabolite is inactive. The one which is active is the one through reduction. Remember, these are sulfoxide, so it can undergo reduction to give us a sulfide. The sulfide is SCH3. So the only thing that has happened between this and that is that this has undergone, the sulfoxide has undergone reduction to give us the sulfide. Then also, so that is S. This is the same structure. What here has happened? That can't see any difference here. So this is glucuronic. There we have glucuronic conjugation. There is the same. There is the same. I think that there is a mistake there. Suppose here there is no difference between this and Sulidac. That's CH2CO2. So, but we are also saying the compound itself, a Sulidac, can also undergo glucuronic conjugation, the same kind of metabolism. So the main routes of metabolism for Sulidac is oxidation to the sulfur conjugation. Of course, the oxidized metabolite can undergo conjugation. Then it can undergo reduction, of course, to give us a sulfide, which is active, or it can undergo glucuronide conjugation without the oxidation. Those are the main routes of metabolism for the sulidac. So very, very similar, of course, because we do not have a nitrogen here anymore. Of course, there cannot be hydrolysis. Mainly glucuronide conjugation, oxidation, and reduction. And reduction indeed is what gives us the active compound. Then the next, which is an indole derivative, again, you can see this is an indole derivative, is a todolac. And as I said last time, look at where the carboxylic group is. And look at the distance between the carboxylic carbon or the carboxylic group with the first or with the first ring of the indole ring. You can see that you can count. This is the carbon, you can count 1, 2. So in a todolac, the carbon of the with 2 carbon atoms from the first. This is our first. This from the first heterocyclic ring with retention of activity. And all we are saying that why that happens is because of the fact that the second carbon is a part. It's a part of this ring. It is a part of a cyclic ring. This carbon is a part of the cyclic ring. That's the cyclopipe with retention of activity. And because of this carbon being retained within this cyclic ring, it's not free. Then that constraint allows for this molecule to still be able to interact with the receptor. This allows this molecule to still interact with the receptor. And you can see, of course, also for this drug, the second hydrophobic group is actually, we do not have a second ring, but we do have, of course, that alkyl group, just as in ibuprofen. So when you look at this drug, please note, although it is classified as an acetic acid derivative, because even when you put this substituent, the chirality here does not have any influence on activity, but it is called a non-classical acetic acid derivative because it does not really agree with an expected SAR, where you would say that separation with a 2 carbon atom abolishes activity found to have non-steroidal anti-inflammatory activity. We are saying it can also be classified as a pyranocarboxylic acid. This is what we call a pyran. That's why we are calling it a pyranocarboxylic acid, a chemical class of anti-inflammatory drugs. It is not strictly an aryl acetic acid derivative because there is a 2 carbon atom separation between the carboxylic acid function and the heteroaromatic ring. So that's, I hope that is very clear now. We cannot say it's a true acetic acid derivative, but the fact that its characteristics in terms of activity resemble the characteristics of the acetic acid derivative. That is why we classify it here. But as I said, it is a non-classical. It still possesses structural characteristics similar to those of the heteroaryl acetic acid derivatives, and that is why it's classified here. The structural characteristics are where we are saying, even after putting the R group to make it chiral, the do not have differences in the activity. There is equipotence, and that is only expected when you have the acetic acid derivative. So it's structural characteristics that make this drug to be classified as an acetic acid derivative, but a non-classical acetic acid derivative. The primary mechanism of action appears to be inhibition of the biosynthesis of prostaglandin, the same, with no inhibition of the repair. Possesses a more favorable ratio of inhibition of prostaglandin biosynthesis in human rheumatoid viocytes and chondrocytes than gastric mucosal cells compared. In all we are saying in these many words is that it has lower GIT irritation compared to ibuprofen, indomethacin, naproxen, diclofenac, and piroxicam cell compared to this drug. So it's kind of a todolac is belongs to the class we had said preferential cyclooxygenase enzyme inhibitors. More preferentially goes for the inducible COX than the constitutive. Thus, although etodolac is no more potent than many others, the lower incidence of GIT side effects represents a potential therapeutic advantage over the others because it has more preferential activity. It does not go for the GIT for the GIT's COX enzyme. It goes mainly, we did mention, the ones no more further not in the synovial fluids in the joint are the prostaglandins that are produced because of the inducible cyclooxygenase enzyme. So it has preferential binding for COX-2 than COX-1, but it is not as selective. It is still non-selective with preferential binding. It is highly bound to plasma proteins expected because, again, of its carboxylic acid penetration. The synovial fluid is greater than that or equal to that of that. Only diclofenac appears to provide greater penetration compared to this drug. It is metabolized to 3-hydroxylated metabolites and glucuronide conjugates, none of which possess important pharmacological activity. So when it comes to metabolism, it's hydroxylated at position 6, 7, and 8. Metabolism appears to be the same in the elderly as in general population, so no dosage adjustment. That's a lot of pharmacology. It is indicated again for the management of the signs and symptoms of arthritis and for management of pain. So all we are saying, again, metabolism through oxidative hydroxylation, 1, 2, 3. So through oxidative hydroxylation, again, all we have again mentioned is the fact that it has a unique structure, but also still fits in this particular in this particular group. Acyl groups at R1 and an acetic acid function, at R2 an enhanced acetic acid inflammatory activity. All we are saying is this is a general structure. So when you look at this general structure, all we are saying is an acyl group at R1, acyl group at R1, and acetic acid function at R2, enhanced acetic acid activity. Remember, have we gone again? So we remember this, one of these groups is an acyl group. One of these groups is the acetic acid derivative. And also, if you look at this, this is also an acyl group. So that's all we are saying. That's a very general structure. We're only saying what these groups require to be. All optimum activity was found when R1 was an acyl group, R2 was an acetic acid function. That was responsible for maximum activity. Increasing the chain length of R1 subsequent to gave derivatives that were 20 times more potent than methyl. That is for R1. All we are saying, and if you look at etodolac, we are only saying, if you look at etodolac, this R1 here is an ethyl group. So all we are saying, if you put a methyl group at this position, there will be reduction in activity. When you increase it to either an ethyl or propyl group, there is better activity. If ethyl gives good activity, just like propyl, there is no need of going to propyl. And that is why it is an ethyl group. So we are saying lengthening, increasing the chain length of R1 to ethyl or N-propyl is better than when it's a methyl. That explains why the group is an ethyl group. Lengthening the acid chain or ester or imide derivatives gave inactive compounds. All we are saying again, very straightforward, that if this acid, if you lengthen it now further, already you have 2 carbon atoms separating it from your first hydrophobic area. You make it longer, lengthen it, derivatize it with an amide, then you lose activity. Some of those points you already said, you require a free carboxylic group to give you the binding site for this drug. So lengthening this acid chain or ester or amide derivatives will be inactive. The corresponding alpha methyl acetic acid derivatives were also inactive. The corresponding alpha methyl acetic acid. So, and that's why we are saying it is behaving more. It is behaving more like the acetic acid derivatives and the we said, if you put, this is the alpha group, you're considering this to be alpha group. For the propionic acid derivatives, if you put a small methyl group there or a small alkyl group there, you expect increase in activity. Of course, you expect chirality with the S isomer being more active. But we are saying for etodolac, if you put a methyl group there, you will just destroy activity. So that is more closer to the acetic acid derivatives than
Amen.
the propionic. A number of substituents in the aromatic ring were evaluated. The substituent at the 8th position were most beneficial. Among the most active were 8-ethyl, 8-N-propyl, 7-fluoro, 8-methyl derivatives. We are only again looking at the substituent. We are trying to explain why a Todorac. In fact, that's what we are asking ourselves. Why a Todorac? Why did they come up with the specific substituents at the specific positions? And we are saying, at again the aromatic ring, that is the ethyl group, N-propyl group. Those are some of the groups that were found to be very active, and that is why at that position you have an ethyl group, because it was found to give us good activity, and therefore no wonder the compound we call it Todorac. We have already explained that one. Again, we said an ethyl or an N-propyl gives us good activity. We explained here you cannot lengthen, you cannot substitute, and that explains the SAR of a Todorac. So we are saying a number, so we can see, yes, we have mentioned 8-ethyl. So you could put substituents at different positions. You could put substituents at different positions. So this is the position 8, 7, 6, 5, 4, 3. So you could put this was the position we're calling on this ring. I'll get to the numbering, the proper numbering. I think it is going 1, 2, 3, 6, 7, 8. Yeah, we are starting here. Then 1, 2, 3, 4, 5, 6. It's 1, 2, 3, 4, 5, 6, 7. It looks like this is what they're including, these positions, because it can only go 1. Or can you start to see that 2, 3, 4, 5, 6? Yeah, I'll get the correct number, but I'm assuming it's 1, 2, 3, 4, 5, 6, 7, 8. So we are saying on this aromatic ring, they try to put substituents. This substituent R3 at different positions, but it was found which was responsible for maximum activity is when that R3 was at this particular position, position number 8. In any other position, it was found to interfere or diminish or drastically reduce or even abolish activity. That's why we are saying 8-ethyl, 8-N-propyl, 7-fluoro, and 8-methyl. So for 7, it must be specifically a fluoro and 8-methyl derivatives. And therefore, that is why ethyl group was found to be most appropriate. Of course, these are done. You put the group. This is how the drug discovery and development. You put the group and then you test for activity, and whichever group is responsible for best of activity with least toxicity, then that is the best group at that position. So this usually you determine through the clinical trials. Todorac was found to possess the most favorable anti-inflammatory to gastric distress properties among these analogs. Concludes it all that all the other substituents which could have been done at the different positions did not give the best molecule. It is those substituents that are found in a Todorac that were responsible for the molecule which had optimum anti-inflammatory activity and minimum toxicity, especially GIT irritation. And that is why when you are, remember I said earlier, when you are discussing that molecule, if you are told to discuss the specific groups within a Todorac, don't start saying this group can be changed to this. You are just explaining why that group at that position. Why do you have the ethyl group at position 8? Why do you have the acetic, unsubstituted acetic group? So you will concentrate when you are discussing the SAR of a specific molecule, you just talk about that molecule without bringing in any other molecule. When you are discussing the SAR of the group, that is when now you start saying this group can be replaced with a fluoro group with increase or decrease in activity. Please let us note, read the question properly and be aware. Are you discussing that molecule by its name, or are you discussing the class of, or the class or the group of those drugs? Like, are you discussing the acetic acid derivative? We are discussing the acetic acid derivative, which would be very wide. We are talking about, you can start with indomethacin, you are going to sulidac and all, and the various groups. But when you are discussing indomethacin alone, you will be discussing those groups which are at certain positions. So now we are done with the first category, which is the indo and indene acetic acid derivatives. We have discussed domethacin, we have discussed Sulidac, and we have discussed Etodorac. Now the next category is the indo-acetic acid derivatives, and in the indo-acetic derivatives, as you can see here, very, very simple, the indo was replaced by a pyrorane. Definitely this is a pyrorane with retention of activity. And if you look at tometin, you can see there is not much change of a structure, not much change of structure. Indeed, it really does agree. It really does agree with the general structure. It does agree with the general structure we had of the non-steroidal anti-inflammatories. There you have your pyrorine. Remember, you're calling them acetic acid because of this group here. It's an acetic acid, the group that's attached to a first hydrophobic group. So you can see it agrees, because remember, we had said even in ketoprofen, which does not have this kind of group, you have that is our first hydrophobic group, and then it is separated from the second hydrophobic group by a polar group, and this polar group is the carbonyl group. Again, this carbonyl group, as we said earlier, it helps us in rigidity and helps us to give us the most active group. Most active group, so that now we do not have many different molecules. So that is tometin. You can see it as an N-methyl, and because of this N-methyl group, remember the N adds electrons, pushes electrons to the methyl group, and therefore this methyl group is highly susceptible to metabolism, and also this methyl group even this methyl group highly susceptible to metabolism, and therefore the derivative which was done, because this methyl group is highly susceptible to metabolism, they replaced that methyl group with a chloro group.
Yeah, so I don't know what happened. I was not in the meeting.
So here, so that is the example there.
I just received the program.
So you look at the tometin, it's related to domethacin, with each containing a non-coplanar para-substituted benzoyl group and function. And you can see that is what it is. If you look at tometin, this is our benzoyl group, but of course for this is toluene. Precisely. This group is toluene. When the phenyl group is substituted with the methyl group, that is toluene. So it's a toluene. The other one was a benzoyl. This is a toluene derivative. So you can see that is one of the similarities. A non-coplanar para-substitute benzoyl group and acetic acid function, and definitely both of them are function. Tometin possesses a pyrorine in place of the indorine in indomethacin, and we are calling it a toluene because it's a toluene. So the 5-para toluene group was replaced with a para-chlorobenzoyl moiety and a methyl group introduced in the 4th position of the pyrorine to give us somepirac. So if you look at somepirac, we are saying that the toluene group was replaced with a benzoyl group. Position 1, 2, 3, 4, there was introduction of a methyl group. Definitely this methyl group, again, very important in pushing this group out of planarity, pushing it out because of the introduction of steric hydrons. But this drug, though with good activity, was also found to have quite some toxicity, and I think it's actually obsolete. So 5-para-toluene group was replaced with a para-chlorobenzoyl moiety and a methyl group introduced in 4th position of the 4-methyl 5-para-chlorobenzoyl analog is approximately 4 times as potent as tometin. Can we be able to explain why this molecule is more active than this? I'd already said this methyl group of toluene highly susceptible to oxidative hydroxylation. That is one thing. Number 2, this group here, introduction of this methyl group in somepirac increases steric hydrons and pushing the benzoyl ring out of planarity with the pyro ring, giving us a better fit for the non-steroidal anti-inflammatory receptor.
Okay.
The paracro group blocked oxidative metabolism, increasing duration of action, of course, in somepirac, as we have already mentioned. So somepirac, it was removed from the market. In other words, it's obsolete because of severe anaphylactic reaction sensitive to aspirin. So although it's better, much activity, don't discuss it much because it was removed from the market. Propionic acid analog is slightly less potent than tometin. Pharmacokinetics. It is rapidly and almost completely absorbed on oral administration. It has a relatively short plasma half-life because of extensive fast-pass metabolism involving hydroxylation of the paramethyl group to the primary alcohol and then subsequent to the dicarboxylic acid. We are calling it dicarboxylic acid because it will be converted to carboxylic acid at the para position, and of course there is also the carboxylic acid that is attached to the pyro ring. The metabolite is inactive. It is highly bound to plasma protein, and excretion of tometin occurs primarily in the urine. That is our tometin. So we are saying mainly this group here oxidized to the carboxylic acid, but it will go through an alcohol. So oxidative hydroxylation and then oxidation, further oxidation to the metabolite, which is inactive.
Okay.
So we are only saying when we look at the tometin, it has structural resemblance with the indoor ring. It has the portions of the acetic acid necessary for activity, the 3 portions of indomethacin deemed necessary for activity, the carboxyl group, the flat indoor ring, and an out-of-plane phenol ring. So it possesses all that, and therefore very comparable to indomethacin. Compared to indomethacin, is there anything to ensure SAR3 that one conformer predominates and the 2 aromatic rings are non-coplanar, which is more potent, and why? It's the same thing we have said. If you look at tometin compared to indomethacin, you do have this fixed bond, which allows mainly for one conformer. So it allows, because of this methyl group, you can see this N-methyl group. Because of this N-methyl group, this group can either be on that side where there is no methyl group, or it can be on this side where there is a methyl group. So it is a bit different. It will allow this group here, will bring about stellic hydrons, and it will push this group that way out of, that way to give us one conformer, which will give us more activity. Also, if you look at this group, this compound, you are saying, if you look at at this position, definitely you have a hydrogen at that position. At this nitrogen, you also have a methyl. So this hydrogen here, and this methyl at that position, very similar to what we had in indomethacin. These 2 groups will push this group, make this ring, because you have a group protruding there, which is a hydrogen. You have this methyl protruding here. These 2 will introduce steric hydrons, and you push this ring out of coplanarity, so that this ring and that ring will be out of coplanarity. So that's the 2 things that we observed with indomethacin. Although the structure looks different, will still be obeyed. We are still saying that tometin retains the partial rigidity introduced by the carbonyl group, and therefore it will allow for existence of one conformer. It will allow for existence of one conformer where this group is much further from the methyl group. So that conformer will be responsible for maximum activity. Because also of the methyl group at that position number 1, and the hydrogen at that position 1, 2, 3, 4, at position number 4, those 2 allow for pushing this second ring, the toluene group, out of planarity, and therefore the tometin will have some structural resemblance with indomethacin to give us maximum activity. Tormenting major metabolite, we have said, is carboxylic acid resulting from benzylic hydroxylation and subsequent oxidation. And of course, as we have said, it has a very, very short half-life.
Yes.
Any question up to there? Oh, it's already 11. Wow, we haven't done much, but at least
actually,
we have done something.
you know, she
Any question?
And she asked me, what does your committee want? How do they want me to? As in, you give her the topic. But I think it's not a problem because we can say the issue of whatever, LGBTQs, whatever. Or the problem,
because we have discussed a lot. I want you just to look at the structure of ketorolac. Look at this, it's only ketorolac we haven't discussed, oxaprozin, and come out with the structural similarities, structural differences between those two drugs, ketorolac and oxaprozin, compared to the drugs that we have already discussed in this class, the acetic acid derivatives, and we will be able to finish off that because of.
But to generally talk about.
Plus, we have left the phenyl acetic acid derivatives, which are not major. So we'll be able to finish that class, and then we move on to the propionic acid derivatives. We are not able to do much, but at least we have done something. And for that, we thank God. It's better than nothing. But I still need us to see whether we can add one or more, one lecture at least from next week. Naomi, is it possible?
No, not really.
We cannot give up. Interruptions come so that we give up, but we not give up. So can we, is it possible for us to add one lecture next week?
Because living, definitely we are living.
Even if it's after 4.
Accommodating that aspect of the
class students, my dear students, can we have an extra class? Even as we think of when to do a fast cut.
Anyway, just think about it, Chair, and I will also think about it. And I know even Penina is going to come up with something, then we can compensate what is good.
Hey, students, you are quiet. What has happened?
Okay.
Can we sign in? Can you sign in? At least we were there. We didn't do much, but we have done something. And for that, I appreciate, because we do not have power. I don't have power in my house. And therefore, whatever we have done, I'm thankful about it. An addition is not a subtraction. What we have done today will not repeat. So it is too positive, as little as it is. Really humbly requesting if we can add one lecture next week.
You
look at your timetable, see whether we can add anything. Even if it's after 4, I will be willing to sacrifice. And then also discuss when we can have our fast cut. Have you started your practicals?
He did it well.
Yes. Yes. Makeup class. Let's see. At least we have done something little, but and I hope you are understanding well, because that's also very important. So you are all following. Have you started on your practicals? All the unit.
Yeah, exactly. Thank you.
Oh, which practical did you do?
Yes.
You did on which drugs?
The challenge,
but I know even this one, because even here we have many practicals you can do. Non-aqueous titration, acid-based titrations, you will be able to do 1 or 2, okay? So kindly discuss, and let's have an extra lecture next week. Continue signing in if you haven't signed in. I hope it was informative. I hope you have learned something. But continue signing in if you haven't. I'll start looking at your assignment next week. I see how well you answered the questions, and I will also look at the notes and see what I can send you. So I'm still organizing to send you a bit of notes, but I need all students to attend. I don't know how many we are supposed to be.
We can
because
say that
today we are only 32. I don't know how many we are supposed to be in this class.
Christian faith. Yeah.
So how many are we today? How many have signed in? 1, 2, 3, 4.